Hello there, and welcome to Zzscience, where the mysteries of life unfold like gentle bedtime stories, and every discovery becomes a quiet path to rest. Tonight, we're wandering into one of nature's most elegant secrets, DNA, that tiny twisted ladder hiding inside every cell in your body, quietly holding the instructions for everything that makes you, you. Before we begin our gentle journey through the spiral corridors of genetics, feel free to share what time it is and where you're listening from. It's always wonderful to know where these quiet explorations are drifting off to. Now, DNA might sound like serious science, and it is, but it's also poetry written in a language older than words, a story that connects you to every living thing that's ever existed. We'll follow its discovery from dusty laboratories to the sparkling double helix, explore how it copies itself with remarkable precision, and discover why this microscopic masterpiece is both the author of diversity and the keeper of continuity. So settle in, let your breathing slow, and let's take a gentle wander through the most personal story ever told, the one written inside you. Picture, if you will, a spiral staircase in a castle made of molecules. Not the grand, sweeping kind you'd find in a palace, but something more intimate. A delicate, twisting ladder so small that millions could dance on the head of a pin. This is DNA, deoxyribonucleic acid, though we can simply call it the most beautiful architecture that life ever invented. The shape is iconic now. Everyone's seen that graceful double helix, two ribbons spiraling around each other like dancers who never tire, never stumble, never miss a step. But for most of human history, this elegant structure was completely invisible, hidden away in the nucleus of every cell, quietly going about the business of being you. If you could shrink yourself down, smaller than the tiniest mite, smaller than the smallest bacterium, and slip inside one of your cells, you'd find yourself in a bustling molecular city. And there, coiled and folded in the nucleus like the world's most organized library, would be your DNA. All 6 billion letters of it, twisted into 23 pairs of chromosomes, each one a masterpiece of compact design. The ladder itself is made of sugar and phosphate, forming what scientists poetically call the backbone. But the real magic happens in the rungs, the steps that connect one side to the other. These aren't made of wood or metal, but of 4 simple chemical letters, A, T, G, and C. Adenine, thymine, guanine, and cytosine, though their names matter less than their endless dance of connection. Here's where it gets quietly astonishing. These letters don't pair randomly. A always reaches for T, and G always embraces C. It's like a molecular romance that never wavers, never cheats, never gets confused about who belongs with whom. This faithful pairing is what allows DNA to copy itself so precisely, and it's why the two sides of the ladder are perfect mirrors of each other. If you could stretch out all the DNA in one of your cells, it would reach about 6 feet, taller than many people, all tucked away in a space smaller than the period at the end of this sentence. It's a marvel of molecular origami, folded and coiled with mathematical precision, and every single cell in your body, all 37 trillion of them, contains this same 6-foot library, perfectly duplicated, perfectly organized. The double helix isn't rigid like a metal spring. It's more like a rope ladder that can twist and bend and breathe. It has to be flexible because it's constantly being read, copied, and repaired. Molecular machines smaller than viruses crawl along its length, reading the sequence of letters like a player piano reading music, translating the code into the proteins that build and maintain your body. There's something peaceful about imagining this molecular ballet happening inside you right now. While you rest, millions of tiny readers are making their way along your DNA, transcribing recipes for healing, growth, and renewal. The same process that's been happening for billions of years, refined and perfected through countless generations, now quietly humming away in your cells as you drift towards sleep. But DNA isn't just beautiful, it's also remarkably stable. The chemical bonds that hold it together are strong enough to preserve information for thousands, sometimes millions of years. Scientists have extracted readable DNA from mammoth bones frozen in Siberian ice, from insects trapped in amber, from ancient seeds that still remember how to grow. It's nature's most durable hard drive, a storage system that makes our best technology look like sand castles at high tide. And yet, for all its stability, DNA is also wonderfully dynamic. It can be modified, edited, repaired. Special enzymes patrol its length like molecular mechanics, fixing breaks and correcting errors. Other proteins help it coil and uncoil at just the right moments. It's a living document, constantly being maintained and updated, never quite the same from moment to moment, yet always fundamentally, recognizably you. The scale of it all is both humbling and magnificent. Each cell nucleus contains more information than the largest libraries ever built, written in a script so efficient that it makes our alphabet look clumsy. 4 letters used in endless combinations, creating the instructions for eyes and hair and heartbeats, for the way you smile and the sound of your laughter. What's perhaps most remarkable is that this same elegant structure, this double helix, this twisted ladder of life, exists in every living thing. The DNA in your cells shares the same basic architecture as the DNA in a rose, a whale, a bacterium, a tree. The language is universal, the grammar identical. Only the sentences change, spelling out the difference between scales and feathers, between roots and wings. So tonight, as you settle into rest, there's something wonderfully comforting in knowing that inside each of your cells, this beautiful molecular sculpture is quietly holding the story of who you are. Not just the obvious parts, your eye color, your height, but subtler things too. The way your cells repair themselves, the rhythm of your circadian clock, even perhaps some echo of your great-grandmother's laugh, encoded in loops and spirals too small to see, but profound enough to shape a life. Science has a way of stumbling upon its greatest treasures when it's looking for something else entirely. Such was the case in 1869, when a young Swiss doctor named Friedrich Michel was studying white blood cell nuclei from pus-soaked bandages collected from a local hospital. Not exactly the most glamorous research material, but Michel was curious about what lurked inside the cell's central command centers. What he found was a strange, phosphorus-rich substance that didn't behave like any protein he knew. He called it nuclein. We now know it as DNA. But Michel had no idea he'd just discovered the molecule that would revolutionize our understanding of life itself. To him, it was simply an interesting chemical curiosity, like finding an unusual pebble on a beach without realizing it was a diamond. For the next 80 years, DNA remained largely a mystery. Scientists knew it existed, they could extract it from cells, but nobody quite understood what it did. Many assumed it was too simple, too repetitive, to carry important information. The real action, they thought, must be happening with proteins, those complex, varied molecules that seemed much more likely to hold life's secrets. It wasn't until the 1940s that DNA began to reveal its true nature. Oswald Avery, Colin MacLeod, and Macklin McCarty performed elegant experiments showing that DNA, not protein, was the transforming principle that could change one type of bacterium into another. They proved that DNA was indeed the carrier of hereditary information, though the scientific community was slow to embrace this radical idea. Around the same time, Erwin Chargaff made a crucial observation. He noticed that in DNA samples from different species, the amount of adenine always equaled the amount of thymine, and the amount of guanine always equaled the amount of cytosine. This became known as Chargaff's rules, and while it seemed like a simple chemical fact, it was actually a vital clue to DNA structure, though no one quite realized it yet. The race to solve DNA structure began in earnest in the early 1950s. It was like a molecular detective story with brilliant minds scattered across England and America, each pursuing different approaches to the same puzzle. Some used chemistry, others turned to physics, and a few employed a relatively new technique called X-ray crystallography, essentially taking photographs of molecules using X-rays instead of light. At King's College London, Rosalind Franklin was becoming the world's expert at photographing DNA. Her X-ray crystallography images were works of art as much as science, beautiful, symmetric patterns that revealed DNA's hidden architecture. Her most famous image, Photo 51, showed clear evidence of a helical structure with the molecules arranged in a spiral pattern. But Franklin was cautious, methodical. She wanted to gather more evidence before drawing conclusions. Meanwhile, at Cambridge University, James Watson and Francis Crick were taking a different approach. They were model builders, trying to work out DNA structure by constructing physical representations using metal plates and wire. They had access to Franklin's data, though the ethics of how they obtained it remains controversial. And they combined her crystallographic evidence with Chargaff's base pairing rules and their own chemical insights. The breakthrough came in February 1953. Watson and Crick realized that if the two strands of DNA ran in opposite directions, what scientists call antiparallel, and if the bases paired specifically, A with T, G with C, then everything clicked into place. The double helix wasn't just beautiful, it was functional. The complementary base pairing meant that each strand contained the information needed to reconstruct its partner. It was, as Crick famously said, so beautiful it had to be right. Their paper, published in Nature in April 1953, was remarkably brief, just over 1 page. The tone was almost understated, ending with the dry observation that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. In scientific terms, this was practically shouting from the rooftops. The discovery sent ripples through the scientific world, though it took time for the full implications to sink in. Here was the secret of heredity, the explanation for how traits passed from parents to children, the molecular basis of evolution itself. Life, it turned out, was written in a code that could be read, copied, and eventually understood. Rosalind Franklin, whose work had been crucial to the discovery, tragically died of ovarian cancer in 1958 at age 37, 4 years before Watson, Crick, and Maurice Wilkins shared the Nobel Prize for the discovery. Franklin's contributions were largely overlooked for decades, a reminder that scientific breakthroughs often involve more minds and more complexity than the simple narratives suggest. But perhaps the most remarkable thing about DNA's discovery was how it opened doors rather than closing them. Each answer led to new questions. How exactly did DNA copy itself? How was its information translated into proteins? How did mistakes in the code lead to disease? The double helix wasn't the end of the story. It was the beginning of an entirely new chapter in our understanding of life. Today, what began as Misha's curiosity about pus has blossomed into fields like genetic engineering, personalized medicine, and evolutionary biology. We can now read DNA sequences as easily as books, edit genes with molecular scissors, and trace the history of life through changes in the genetic code. We've learned that humans share about 99.9% of their DNA sequence with each other, and surprisingly large percentages with other species, about 96% with chimpanzees, 60% with fruit flies, even 50% with bananas. The story of DNA's discovery reminds us that science is fundamentally a human endeavor, driven by curiosity, collaboration, and occasionally productive rivalry. It's built on countless small observations, patient experiments, and sudden flashes of insight. And sometimes, just sometimes, it leads us to truths more beautiful and profound than we ever imagined when we first started looking. Imagine you had to write the entire encyclopedia of human knowledge using only 4 letters. Not the full alphabet, mind you, just A, T, G, and C. It sounds impossible, doesn't it? And yet, that's exactly what DNA does, every day, in every living cell. With just those 4 chemical letters, arranged in endless combinations, nature has written the instructions for everything from the blue of your eyes to the beat of your heart. This is the language of life. And like any language, it has grammar, vocabulary, and syntax. But unlike human languages, which evolved to communicate between minds, DNA's language evolved to communicate between generations, to pass the essential instructions of life from parent to child, across millions of years and billions of cell divisions. The alphabet is elegantly simple. A for adenine, T for thymine, G for guanine, C for cytosine. 4 chemical compounds that serve as DNA's letters. But simplicity can be deceptive. With 4 letters, you can create 16 different 2-letter combinations, 64 different 3-letter words, and 256 different 4-letter sequences. By the time you're working with longer sequences, the possibilities become virtually infinite. Here's where DNA's language gets particularly clever. It's written in words exactly 3 letters long. These 3-letter combinations, called codons, are the basic units of genetic meaning. Each codon codes for a specific amino acid, the building blocks of proteins. AUG always means start here, while UAA, UAG, and UGA all mean stop. It's like punctuation, but for molecules. The beauty of this system lies in its redundancy. There are 64 possible 3-letter combinations, but only 20 common amino acids, plus a few start and stop signals. This means that multiple codons can code for the same amino acid, what scientists call the genetic code's degeneracy, though there's nothing degenerate about it. This redundancy is actually protection, a built-in error correction system that helps ensure small changes in the DNA sequence don't necessarily change the final protein. Think of it like having multiple words that mean the same thing. If you misspell beautiful as beautiful, you might still understand the intended meaning. Similarly, if a DNA sequence changes from GAA to GAK, both still code for the same amino acid, so the final protein remains unchanged. It's a remarkably forgiving language, considering how much depends on getting it right. But DNA doesn't just store information, it actively participates in reading itself. When a cell needs to make a particular protein, it doesn't directly use the DNA as a template. Instead, it creates a working copy using a related molecule called RNA. Think of DNA as the master cookbook stored safely in the library, while RNA is the photocopied recipe you take into the kitchen. This process, called transcription, is like having a molecular scribe carefully copying selected passages from the DNA library. An enzyme called RNA polymerase slides along the DNA, reading the sequence of letters and creating a complementary RNA copy. But here's a crucial detail. RNA uses a slightly different alphabet. Instead of thymine, T, it uses uracil, U. So when the DNA says A, the RNA writes U, and when the DNA says T, the RNA writes A. Once the RNA copy is complete, it travels out of the cell nucleus to the protein-making factories called ribosomes. Here, the real translation begins. Literally, translation, from the language of nucleic acids to the language of proteins. The ribosome reads the RNA sequence 3 letters at a time, matching each codon to its corresponding amino acid. It's a bit like a molecular assembly line, but one with remarkable precision. Transfer RNA molecules, each carrying a specific amino acid, arrive at the ribosome in the exact order specified by the genetic code. As each amino acid is added, the growing protein chain folds into its proper 3-dimensional shape, guided by the chemical properties of its component parts. What emerges from this process is nothing short of miraculous. A functional protein, crafted according to instructions written in the DNA billions of years ago, but executed with split-second timing in your cells right now. These proteins become everything from the enzymes that digest your food to the antibodies that fight infections, from the collagen that gives strength to your skin to the hemoglobin that carries oxygen in your blood. The language of DNA is also remarkably universal. The same genetic code that operates in your cells works in bacteria, plants, fungi, and virtually every other living thing. This universality is one of the strongest pieces of evidence for the common origin of all life. We all speak the same molecular language, even if we've used it to write very different stories. But like any language, DNA's code has evolved subtle dialects. Some organisms use slightly different versions of the genetic code, particularly in their mitochondria, the energy-producing organelles that may have once been independent bacteria. These variations are like regional accents in the universal language of life, adding complexity without breaking the fundamental communication system. Perhaps most remarkably, DNA's language includes instructions not just for making proteins, but for controlling when and where those proteins are made. Regulatory sequences act like molecular switches, turning genes on and off in response to cellular needs. Some sequences work like volume controls, adjusting how much protein is produced. Others function like timers, ensuring that certain genes are only active at specific stages of development. This regulatory grammar is what allows the same DNA sequence to create such dramatically different cell types. Your liver cells and your brain cells contain identical DNA, but they express different subsets of genes, creating their specialized functions. It's like the same sheet music being played by different instruments. The notes are the same, but the melodies are wonderfully distinct. As you settle into sleep tonight, this ancient language is being spoken fluently in every one of your cells. Genes are being transcribed, proteins are being assembled, cellular repairs are being carried out, all according to instructions written in a 4-letter alphabet that has been refined by evolution for billions of years. It's a conversation between past and present, between the wisdom of ancestry and the needs of the moment, conducted in whispers too small to hear, but profound enough to sustain the miracle of your existence. Right now, as you rest, billions of your cells are performing biology's most spectacular magic trick. Your DNA performs a feat of molecular choreography so precise, so elegant, that it makes the most complex dance look clumsy by comparison. It copies itself perfectly. All 6 billion letters duplicated with an accuracy that would make the world's best printers weep with envy. This process, called replication, might be the most important dance in the universe. Without it, life would have ended before it began. No growth, no healing, no you. Picture this happening right now in your body. Cells in your skin are dividing to replace the ones you've shed today. Blood cells are being born in your bone marrow. Your immune system is creating new defenders against tomorrow's challenges. And each time a cell splits in two, your DNA must first duplicate itself so both daughter cells receive a complete copy of life's instructions. The dance begins with a problem. DNA exists as that famous double helix, 2 strands wound around each other like lovers' hands. But to copy it, the cell must first unwind those strands, separate them. This requires special enzymes called helicases, molecular machines that work like tiny crowbars, prying apart the double helix and creating what scientists call a replication fork. The name is perfect. It looks exactly like a fork in a road, where one path becomes 2. But here's where things get beautifully complex. As the helicase unwinds the DNA, it creates tension further down the molecule. Imagine trying to untwist a rope while someone holds the other end. The twisted part gets tighter and tighter. DNA solves this with another enzyme called topoisomerase, which acts like molecular scissors, making temporary cuts to relieve the tension, then sealing them back up. Snip, release, seal. Snip, release, seal. Once the strands are separated, the real copying begins. Here comes DNA polymerase, perhaps the most remarkable enzyme in biology. It slides along each separated strand, reading the sequence of letters and building a complementary copy. Where it finds an A, it adds a T. Where it sees a G, it places a C. The base pairing rules discovered by Chargaff become the grammar of replication. But DNA polymerase has a quirk. It can only work in one direction, from what chemists call the 5' end to the 3' end. This creates an elegant problem. Since the 2 strands of DNA run in opposite directions, replication can't proceed smoothly on both strands simultaneously. The solution is ingenious. One strand, called the leading strand, is copied continuously as the replication fork opens, smooth and steady, like writing a sentence from left to right. But the other strand, the lagging strand, must be copied in short segments, each running backwards relative to the fork's movement. These segments, called Okazaki fragments after the Japanese scientists who discovered them, are later stitched together by another enzyme called DNA ligase. It's like trying to copy a book where half the pages read normally and half read backwards. Somehow, the cell manages both tasks simultaneously with mechanical precision that never falters. The speed of this process is breathtaking. In human cells, DNA polymerase can add about 50 nucleotides per second. That might not sound fast until you consider that some bacteria can replicate their entire genome in just 20 minutes. Imagine copying a library's worth of information in the time it takes to walk to the corner shop. Yet speed means nothing without accuracy. DNA polymerase doesn't just add nucleotides. It proofreads its work. Like an obsessive editor, it checks each letter as it's added. If it finds a mistake, the wrong base paired in the wrong place, it backs up, removes the error, and tries again. This proofreading reduces mistakes to roughly 1 error per 10 billion letters copied. But even that's not enough. Cells have backup systems upon backup systems. Special repair enzymes patrol the newly copied DNA, scanning for errors that escape the polymerase's attention. They can fix mismatched bases, remove damaged sections, even repair breaks in the DNA backbone. It's quality control taken to molecular extremes. Think about this for a moment. Every time one of your cells divides, it successfully copies 3 billion base pairs of DNA with an error rate lower than 1 mistake per cell division. Most of the time, when errors do slip through, they become mutations, changes that can be harmful, beneficial, or simply neutral. These mutations are evolution's raw material, the tiny variations that allow life to adapt and change over generations. The replication process is so fundamental that it's been conserved across all life. The same basic mechanism that copies your DNA also works in bacteria, plants, and every other living thing. The molecular players have different names, slightly different structures, but the essential dance remains the same. It's been performing this choreography for billions of years, refined through endless generations into the masterpiece we see today. Perhaps most remarkably, this entire process happens in near-perfect synchrony across your body. When your cells decide it's time to divide, they don't just randomly start copying DNA. They wait for the right signals, check that conditions are appropriate, and then begin the process in carefully orchestrated waves. It's like a molecular symphony, with billions of performers all following the same conductor. As you drift towards sleep tonight, this ancient dance continues in your cells. DNA strands unwind, polymerases slide along their tracks, and new copies emerge with clockwork precision. The same process that's been preserving and transmitting life's instructions since the first cells appeared on Earth now quietly maintains the genetic heritage that makes you uniquely you. If you could shrink down to the size of a molecule and take a tour through one of your cells, you'd find yourself standing before the most remarkable library ever assembled. No towering shelves of books, no dusty volumes bound in leather. Instead, coiled and folded in the cell's nucleus, you discover 6 feet of DNA containing more information than has ever been written, stored in a space smaller than the head of a pin. This is your genome, your complete genetic instruction manual. 3 billion base pairs arranged in precise order, containing roughly 20,000 genes, the recipes for proteins that build and maintain your body. It's a library that puts Alexandria to shame, written in the smallest possible print using only 4 letters. But here's what makes this library truly extraordinary. It exists in nearly every cell in your body. Your skin cells carry the same complete library as your brain cells. Your heart muscle contains identical instructions to your liver. 37 trillion cells, each one housing the entire collection. It's as if nature decided that every room in the house should contain a complete encyclopedia, just in case. The scale of information storage is staggering. If you printed out your DNA sequence using normal font, single-spaced, it would fill roughly 3 million pages. Stack those pages, and you'd have a tower taller than the Washington Monument. And that's just one copy. Remember, you have 2 copies of each chromosome, one from mum, one from dad. So double that tower. Yet for all its vastness, your genome is surprisingly compact. Those 6 feet of DNA are folded with origami-like precision, coiled around special proteins called histones, then coiled again and again until everything fits snugly inside the nucleus. It's like packing a mile of rope into a tennis ball while keeping every inch accessible and organized. The organization itself is a marvel of molecular architecture. Your DNA is divided into 23 pairs of chromosomes, each one a massive molecule containing hundreds or thousands of genes. Think of chromosomes as different volumes in the library, each devoted to particular subjects. Chromosome 1 is the largest, containing over 4,000 genes. The tiny Y chromosome, found only in males, contains fewer than 200. Within each chromosome, genes aren't randomly scattered like books thrown on a shelf. They're arranged in neighborhoods, with related genes often clustered together. Some regions are gene-rich, packed with instructions for important proteins. Others are gene-poor, containing long stretches of what was once dismissively called junk DNA, but we now know serves important regulatory functions. Your genome tells the story of your ancestry in ways that would make genealogists weep with joy. Embedded in those 3 billion letters are traces of ancient migrations, evolutionary adaptations, and genetic gifts passed down through countless generations. You carry DNA from Neanderthals, remnants of viral infections that struck your ancestors millions of years ago, and genetic variants that helped humans survive ice ages and plagues. Some of your genes are ancient beyond imagination. The genes that control your basic cellular functions, how to make energy, how to repair DNA, how to divide, are recognizably similar to genes found in bacteria that lived billions of years ago. Other genes are relatively recent additions, acquired as human ancestors evolved bigger brains, more dexterous hands, and the ability to walk upright. But your genome isn't just a static archive. It's a living document, constantly being read, interpreted, and occasionally edited. At any given moment, only a small fraction of your genes are active. The rest wait quietly in the nucleus, like books sitting unread on library shelves, ready to be opened when needed. Different cell types read different sections of the library. Your liver cells primarily access genes for metabolic enzymes and detoxification proteins. Your immune cells focus on genes for antibodies and inflammatory responses. Your neurons emphasize genes for neurotransmitters and electrical signaling. Same library, different reading lists. The timing of gene expression is equally important. During development, genes switch on and off in precisely choreographed sequences. First, the genes for basic body structure, then those for organ formation, then specialization into different cell types. It's like reading chapters of the instruction manual in exactly the right order to build something as complex as a human being. Environmental factors can influence which genes are read and when. Stress, diet, exercise, even social interactions can alter gene expression patterns. This field, called epigenetics, reveals that your genome isn't your destiny. It's more like a piano with 20,000 keys, and life experience determines which melodies get played. Perhaps most humbling is how much of your genome remains mysterious. We can read the sequence, we know where most genes are located, but we're still learning what many of them actually do. Some genes seem to have multiple functions, others work only in combination with distant partners, and still others appear to do nothing at all, though evolution rarely keeps true junk around for long. Your genome also contains its own repair manual. Scattered throughout the DNA are sequences that encode proteins specifically designed to maintain and fix the genome itself. It's self-referential in the most elegant way. DNA that contains instructions for fixing DNA. Genes that encode the very enzymes that copy genes. The uniqueness of your personal genome is almost incomprehensible. While humans share 99.19% of their DNA sequence, that remaining 0.1% represents about 3 million differences. These variations, single letter changes, small insertions and deletions, larger rearrangements, make you genetically distinct from every other person who has ever lived, with the exception of identical twins. Yet for all its complexity and uniqueness, your genome is also profoundly universal. The same four-letter alphabet, the same genetic code, the same basic mechanisms that operate in your cells work in every living thing on Earth. Your DNA shares deep similarities with the genome of every plant, animal, and microbe. We're all variations on the same ancient theme, all reading from editions of the same fundamental book of life. Tonight, as your body settles into rest, this molecular library continues its quiet work. Genes switch on and off in circadian rhythms. DNA repair enzymes fix the day's accumulated damage, and growth factors orchestrate the renewal of tissues worn down by life's activities. Your genome doesn't sleep. It simply shifts into its nighttime reading list, focusing on repair, restoration, and dreams of tomorrow. You have your father's eyes and your mother's laugh, but the story of how you came to possess these traits begins with one of biology's most intimate moments: the creation of an egg and a sperm, each carrying exactly half of their parents' genetic inheritance. This process, called meiosis, is evolution's greatest gambling strategy. Instead of simply copying DNA like other cell divisions, reproductive cells engage in a complex genetic shuffle that ensures no two offspring are ever identical. It's a molecular casino where the house always wins because the house is diversity itself. Picture what happens in your parents' bodies as they create the gametes that would eventually become you. Each parent starts with cells containing 2 copies of every chromosome, one inherited from their mother, one from their father. But to create reproductive cells, something remarkable must occur. These chromosome pairs must separate, with each egg or sperm receiving only 1 copy of each chromosome. But before they separate, something magical happens. The paired chromosomes embrace, literally wrapping around each other in a process called crossing over. During this intimate molecular dance, they exchange pieces of DNA. Your mother's chromosome 1 might swap a section with your father's chromosome 1, creating new combinations of genes that neither grandparent possessed. It's like shuffling two decks of cards together, but instead of creating chaos, it creates endless possibilities. Each crossing over event generates novel genetic combinations, ensuring that the gametes your parents produced were genetically unique. The chromosome you inherited from your mother contains a mosaic of DNA from both your maternal grandmother and grandfather, stitched together in a pattern that has never existed before and will never exist again. This genetic recombination explains why siblings can be so different despite sharing the same parents. Each egg and sperm is a unique lottery ticket, carrying a random selection of ancestral genes. When fertilization occurs, two lottery tickets combine to create something entirely new, you. The mathematics of inheritance are staggering. Humans have 23 pairs of chromosomes, which means each parent can produce over 8 million genetically distinct gametes through chromosome shuffling alone. When you factor in crossing over, the number of possible genetic combinations becomes virtually infinite. Even conservative estimates suggest that human parents could theoretically produce more unique offspring than there are atoms in the observable universe. Yet within this randomness lies beautiful patterns of inheritance. Some traits follow simple rules discovered by Gregor Mendel in his pea garden over a century ago. Brown eyes tend to dominate over blue eyes. The ability to roll your tongue is usually inherited as a single gene. Some genetic diseases follow predictable patterns, passed from parent to child according to mathematical probabilities that geneticists can calculate with remarkable precision. But most human traits are far more complex. Height, intelligence, personality, disease susceptibility. These characteristics result from the interaction of hundreds or thousands of genes, each contributing a small effect. It's like a vast orchestra where every instrument plays softly, but together they create the symphony of human diversity. Environmental factors add another layer of complexity to the inheritance story. The genes you inherit create your potential, but how that potential is expressed depends on everything from nutrition during pregnancy to social experiences in childhood. Your DNA provides the score, but life experience conducts the performance. Some inherited traits skip generations, lying dormant until the right genetic combination awakens them. Others appear unexpectedly, arising from new mutations that occur during egg or sperm formation. Every human carries dozens of new genetic variants that neither parent possessed. Tiny changes that add fresh notes to evolution's endless composition. The story of inheritance extends far beyond individual traits to encompass entire evolutionary histories. Embedded in your DNA are genetic signatures of humanity's journey out of Africa, the agricultural revolution, and countless other chapters in our species' past. You carry genes that helped your ancestors survive ice ages, resist diseases, and adapt to new environments across the globe. Your mitochondrial DNA tells an especially intimate story of maternal inheritance. These tiny powerhouse organelles in your cells contain their own genetic material, passed exclusively from mother to child. Your mitochondrial genes are identical to your mother's and hers to her mother's, creating an unbroken chain of maternal inheritance, stretching back thousands of generations. It's like a genetic family tree written exclusively in the maternal line. The Y chromosome tells a parallel story for males, passed unchanged from father to son, except for occasional mutations that serve as molecular timestamps. These paternal lineages allow geneticists to trace human migrations and family relationships across vast spans of time and geography. Modern genetics has revealed inheritance patterns that would have seemed like magic to earlier generations. We now know that identical twins, despite sharing the same DNA, can develop differently due to epigenetic changes, chemical modifications that alter gene expression without changing the underlying sequence. Environmental factors can literally reshape how genes are read, and some of these changes can be passed to offspring. Perhaps most remarkably, we're discovering that experiences can influence inheritance in ways that challenge traditional thinking about genetics. Trauma, diet, stress, and other environmental factors can alter sperm and egg cells in ways that affect offspring. It's as if parents can pass along not just their genes, but also molecular memories of their experiences. The thread of inheritance connects you to an unbroken chain of successful reproduction stretching back to the first living cells. Every one of your ancestors, for billions of years, successfully found a mate, reproduced, and passed their genetic legacy forward. You are the current endpoint of the longest success story in the history of Earth. Tonight, as you rest, consider the remarkable genetic lottery that created you. The precise combination of chromosomes, the specific pattern of crossing over, the exact moment when one particular sperm met one particular egg, all of these had to align perfectly for you to exist. The odds against your specific genetic combination are so astronomical that your very existence borders on the miraculous. You are simultaneously the product of ancient genetic wisdom, accumulated over billions of years, and a completely novel genetic experiment that has never been tried before. You carry the inheritance of countless ancestors, while representing something entirely new under the sun. In the grand story of life, you are both an echo of the past and a whisper of the future, connected to all life through the elegant thread of DNA inheritance. DNA has a confession to make. It's not perfect. Despite all those proofreading mechanisms, all that molecular quality control, mistakes happen. Letters get swapped, sequences get duplicated, entire chunks sometimes go missing. These errors, called mutations, might sound like cellular failures, but they're actually evolution's greatest gift to life. Without mutations, you wouldn't exist. Neither would roses or whales or bacteria. Earth would still be populated by identical copies of the first self-replicating molecules, frozen in biological amber for billions of years. Change, it turns out, is not just inevitable. It's essential. Most mutations are tiny affairs. A single letter change here, a small deletion there. Imagine retyping a massive encyclopedia and occasionally hitting the wrong key. Sometimes these typos change nothing important, like changing color to color. The meaning remains clear. Other times they create nonsense, like changing cat to CXT. And very rarely they accidentally improve the text, creating something better than what came before. The scale of mutation in your own body is both humbling and astounding. Every time one of your cells divides, it introduces roughly 1 to 3 new mutations into its DNA. Multiply that by the billions of cell divisions happening in your body every day, and you're looking at millions of tiny genetic changes occurring constantly. Most are harmless, repaired by cellular maintenance crews or occurring in non-critical regions. But some persist, creating a slow-motion genetic evolution happening inside you right now. Your genome is actually a patchwork of mutations accumulated throughout your lifetime. The skin cells on your left hand have slightly different DNA than those on your right. Your liver carries mutations that your heart doesn't possess. You're not genetically identical to yourself. You're more like a gentle mosaic of related genetic variants, all descended from the original embryonic cells that began your development. But the mutations that really matter for evolution are those that occur in reproductive cells, the eggs and sperm that pass genes to the next generation. Each human baby is born with roughly 60 new mutations not present in either parent. 60 chances for evolution to try something new. Most will have no effect, some might be harmful, but a few might prove advantageous in ways we can't predict. These germline mutations are how species change over time. They're the raw material of evolution, the genetic variation that natural selection acts upon. In populations, beneficial mutations slowly become more common, while harmful ones are gradually eliminated. It's a process so slow, it's usually invisible within human lifetimes, but over millions of years, it transforms single-celled organisms into redwood trees and blue whales. Sometimes mutations work in clusters, creating more dramatic changes. Gene duplications can provide backup copies that are free to evolve new functions. Chromosomal rearrangements can bring distant genes into new relationships, creating novel regulatory networks. Whole genome duplications, though rare and usually fatal, occasionally create evolutionary opportunities by doubling the genetic raw material available for innovation. Human evolution bears the fingerprints of countless mutations that proved their worth over generations. The genetic changes that allow some people to digest milk as adults arose independently in different populations, spreading because they provided nutritional advantages. Mutations that confer resistance to malaria, though sometimes causing other health problems, persist in regions where the disease is common because the benefits outweigh the costs. More recent mutations tell stories of human adaptation to new environments and lifestyles. High-altitude populations carry genetic variants that help them thrive in low-oxygen environments. Some Arctic populations have mutations that help them metabolize high-fat diets. Urban populations may be evolving resistance to air pollution and infectious diseases. But evolution isn't just about survival of the fittest in tooth and claw terms. Sexual selection drives many evolutionary changes, favoring traits that improve mating success rather than survival. The peacock's elaborate tail is evolution's response to female preferences, not environmental pressures. Human evolution, too, has been shaped by sexual selection, influencing everything from our hairlessness to our capacity for music and art. Mutations also explain the gorgeous diversity of life around us. The difference between a hummingbird and an eagle, between a rose and an oak tree, between a human and a chimpanzee, all trace back to accumulated mutations over millions of years. Every species is a unique experiment in genetic variation, shaped by mutations and molded by natural selection. The molecular mechanisms of mutation are as diverse as their effects. UV radiation from the sun can damage DNA, creating specific types of changes. Chemical mutagens can alter bases in ways that lead to mispairing during replication. Even the normal process of DNA copying occasionally stutters, creating repeated sequences or deletions. Some organisms have evolved to be mutation machines, deliberately increasing their mutation rates when stressed. Bacteria facing antibiotics sometimes ramp up their mutation systems, gambling that random changes might provide resistance. Cancer cells exploit similar mechanisms, evolving drug resistance through accelerated mutation, It's evolution in fast forward, adaptation measured in days rather than generations. Your own body contains evolutionary experiments happening in real time. Your immune system deliberately mutates the genes for antibodies, creating billions of variants to test against new pathogens. The immune cells that produce the best antibodies are selected and multiplied, while the rest are discarded. It's Darwinian evolution playing out inside you, creating custom-designed molecules to fight specific threats. Mutations also reveal the deep connections between all living things. The genetic changes that separate humans from other primates are surprisingly small. Less than 2% of our DNA differs from chimpanzees. Yet that small percentage, accumulated over millions of years of separate evolution, has created the profound differences between species. Tonight, as you settle into sleep, mutations are occurring in your cells. Most will be quickly repaired or prove harmless. But somewhere in your body, a few genetic changes are taking hold, adding their whispers to the long conversation between stability and change that defines life itself. You are both the product of ancient mutations that enabled your ancestors to thrive and the ongoing subject of new mutations that might influence future generations. In your DNA lies the story of life's greatest adventure, the endless dance between preservation and innovation, between genetic memory and genetic possibility, between what has been and what might yet become. Mutations remind us that perfection isn't the goal of life. Adaptation is. Change is not life's enemy, but its most faithful companion, quietly ensuring that life remains flexible enough to face an uncertain future with endless creativity and hope. For most of human history, DNA was like a book written in an unknown language, its pages tantalizingly present, but utterly unreadable. We knew the book existed. We could see its elegant structure. We understood it contained the instructions for life itself. But the actual words, the specific sequences of A's, T's, G's, and C's that spell out the difference between brown eyes and blue, between health and disease, those remained locked away in molecular silence. Then, in the late 20th century, scientists developed the tools to finally read life's instruction manual. Letter by letter, gene by gene, we began the slow work of deciphering the genetic code. What we discovered was more beautiful, more complex, and more surprising than anyone had imagined. The journey began with painstaking techniques that could sequence only short stretches of DNA at a time. Early researchers spent years determining the order of just a few hundred letters. Frederick Sanger, one of the pioneers of DNA sequencing, used methods that were as much art as science, involving radioactive labels, gel electrophoresis, and photographic film. It was like learning to read by examining one letter at a time through a magnifying glass. But technology accelerated the process with breathtaking speed. Automated sequencing machines could read thousands of letters per day, then millions. What once took years could be accomplished in weeks, then days, then hours. The Human Genome Project, launched in 1990, with the audacious goal of reading all 3 billion letters of human DNA, was initially expected to take 15 years. It finished in 13, ahead of schedule and under budget. The completion of the first human genome sequence in 2003 was like finally translating an ancient manuscript that held the secrets of human existence. Scientists could now read the genetic instructions for making hemoglobin, insulin, and thousands of other proteins. They could identify the genes responsible for inherited diseases, trace the evolutionary relationships between species, and begin to understand how genetic variation contributes to human diversity. But reading the genome was just the beginning. Like translating a book from a foreign language, knowing the words doesn't automatically reveal their meaning. Scientists discovered that the human genome contains many surprises that challenge their assumptions about how genes work. First surprise: humans have far fewer genes than expected. Early estimates suggested we might have 100,000 genes or more. The actual count is around 20,000, roughly the same as a simple nematode worm. Clearly, genetic complexity isn't just about the number of genes, but how they're used, regulated, and combined. Second surprise: most of the genome doesn't code for proteins. Only about 2% of human DNA consists of protein-coding genes. The rest was initially dismissed as junk DNA. But we now know that much of it serves important regulatory functions, controlling when and where genes are turned on and off. Third surprise: alternative splicing allows a single gene to produce multiple proteins. Human cells can edit the initial RNA transcript, cutting and pasting different sections together to create protein variants. It's like having a recipe that can be modified to create entirely different dishes depending on which ingredients you include. Modern DNA sequencing has become so fast and affordable that it's revolutionizing medicine, agriculture, and our understanding of life itself. A complete human genome sequence that cost $3 billion in 2003 can now be obtained for under $1,000. Soon, genome sequencing might be as routine as blood tests. This technological revolution has spawned entirely new fields of science. Personalized medicine uses genetic information to tailor treatments to individual patients. Pharmacogenomics studies how genetic variants affect drug responses, helping doctors choose the right medication at the right dose for each person. Gene therapy attempts to treat diseases by introducing correct copies of faulty genes. Scientists can now compare genomes across species with unprecedented detail. They've sequenced the DNA of Neanderthals from bones tens of thousands of years old, revealing that modern humans carry traces of ancient interbreeding. They've traced the genetic changes that allowed humans to develop language, larger brains, and the ability to digest new foods. Agricultural genomics is transforming how we grow food. Scientists can identify genes that confer disease resistance, improve nutritional content, or help crops adapt to climate change. They can trace the evolutionary history of crop plants, understanding how thousands of years of human selection shaped the foods we eat today. The ability to read DNA has also revolutionized forensic science, archaeology, and conservation biology. Tiny traces of genetic material can identify crime suspects, reveal the contents of ancient diets, or help protect endangered species by understanding their genetic diversity. But perhaps the most profound impact of genome sequencing is what it's teaching us about ourselves. We've learned that human genetic diversity is far greater within populations than between them, undermining simplistic notions of race. We've discovered that we share genes with every living thing on Earth, from bacteria to blue whales, revealing our deep kinship with all life. Genome sequencing has also revealed the extent to which our genetics are shaped by our microbial companions. The human microbiome, the collection of bacteria, viruses, and other microbes that live in and on our bodies, contains more genetic material than our own cells. We're not just human. we're walking ecosystems, and our health depends on maintaining the right balance of microbial partners. As sequencing technology continues to improve, we're entering an era of real-time genomics. Scientists can now sequence DNA as it's being extracted from a sample, providing immediate results. Portable sequencers can identify pathogens during disease outbreaks, analyze environmental samples in the field, or even sequence DNA in space stations. The future promises even more remarkable developments. Single-cell sequencing can reveal how individual cells differ from each other within the same organism. Long-read sequencing can capture complete gene sequences, including regions that were previously difficult to analyze. Epigenetic sequencing can identify chemical modifications that affect gene expression without changing the underlying DNA sequence. Tonight, as you rest, know that the genetic code that defines you has finally been made readable by human ingenuity. The same 3 billion letters that specify your eye color, your height, and your susceptibility to certain diseases can now be read as easily as the words on this page. We've learned to speak the language of life itself, and that conversation is just beginning to reveal its secrets. And so, we arrive at the end of our gentle journey through the spiraling corridors of life itself. We've wandered from the molecular architecture of the double helix to the vast libraries contained in every cell, from the elegant dance of replication to the quiet mysteries that still elude our understanding. DNA, it turns out, is far more than just an instruction manual. It's a poem written in chemistry, a story that connects you to every living thing that has ever existed, a thread woven through time that links the first flickering spark of life to your own beating heart. In its twisted ladder lie the secrets of inheritance and change, of memory and possibility, of what has been and what might yet become. Tonight, as you settle into sleep, remember that you carry within you 3 billion years of evolutionary wisdom, carefully preserved and passed down through countless generations. Every breath you take, every dream that dances through your mind, every cell that quietly repairs itself while you rest, all of it orchestrated by that elegant molecular symphony playing out in the nucleus of every cell. And if you found any wonder in this quiet exploration, if the mysteries of genetics brought you peace, or if the questions helped you drift gently toward your dreams, feel free to follow us for more gentle wanderings through science in the future. Sleep well, knowing that you are both ancient and entirely new, both a continuation of life's oldest story and a completely original chapter that has never been written before. Thanks for joining us here at Sleep Science.